Hello, my name is Jessie Wright, and the topic of my video project is chromosomal and DNA mutations. First of all, what is a mutation? According to the genetics lecture number 19 from April 5th, a mutation is defined as a change from one hereditary state to another. To me, that is still difficult to understand, so I made my own definition. A mutation can be defined as a change in a genetic material. Now, genetic material is kind of broad for a definition, so let's examine this a little further. Mutations can occur on chromosomes as well as on individual nucleic acids. Let's start with the nucleic acids. Do you remember what they are? There are 4 of them: adenine, thymine, cytosine, and guanine. A nifty way I use to remember which nucleic acids pair with each other is with the saying, "There's an apple, adenine, in the tree, thymine, next to the car, cytosine, in the garage, guanine." This can be helpful when checking to see if all the nucleic acids are paired correctly. However, mistakes do happen, and these are known as DNA mutations. 3 examples of DNA mutations are substitutions, insertions, and deletions. These are called point mutations because only a single nucleic acid, also known as a base, is changed. First, let's look at substitutions. This occurs when a nucleic acid is paired incorrectly. For example, I have drawn a short DNA template strand with all the bases paired correctly. Here's the same strand, but with a substitution mutation. Typically, this won't cause a problem, but sometimes a substitution can cause a wrong coding for the mRNA and then cause a mistake for the amino acid sequence. This can lead to the production of a malfunctioning protein. A second type of mutation is an insertion. This is when a base is added to the nucleic acid sequence, causing what is known as a frame shift mutation. Originally, the bases would be read in this pattern. But once a base is added, it shifts the way bases are read. This can cause some major issues with the resulting protein, but will most likely render the protein non-functional. The third type of point mutation is called a deletion. It has the same effects as an insertion, except that one nucleic acid base is removed from the sequence. This can also cause some major issues, but will also most likely render the protein non-functional. Now let's move on to chromosomal mutations. What is a chromosomal mutation? It is a change in the number and structure of a chromosome. There are 4 main types of chromosomal mutations: duplication, deletion, inversion, and translocation. First, let's expand on duplication. Chromosomal duplication occurs when extra copies of the chromosome are generated. So here's an illustration of this mutation. Here you can see a chromosome with a pink and orange band on its Q arm. The second chromosome is affected by duplication, and you can see that the pink and orange bands have been repeated once. The second chromosomal mutation is deletion. Chromosomal deletion occurs when some of the genetic material from the chromosome breaks off. Again, here's an illustration of this concept. Here you can see a chromosome with its orange band on its Q arm. Next to it is a mutated chromosome with the orange band deleted. This results in a shortened Q arm. The third type of chromosomal mutation is an inversion mutation. This occurs when a broken chromosome segment gets inversed and put back into place. This illustration demonstrates this concept. Here you can see the same chromosome as before, with its pink and orange band. However, over here is a mutated chromosome, with the pink and orange band reversed. The last chromosomal mutation is called a translocation mutation. This occurs when a fragment from one chromosome breaks off and reattaches to another chromosome.